So last class we have discussed mango up to variety, that the variety grown in different region of the country. The variety, mango variety, as you know, India is the largest mango growing country in the world. Hence, you will find hundreds of varieties are cultivated in Indian subcontinent, out of which some of the important cultivars are cultivated commercially, right? And we have mentioned, we have discussed only those commercially cultivated mango variety which are most popular in the markets, has high demand in the market. So they have high demand in the markets. So, and this, depending upon the climatic situation, there are 4 regions of mango growing in India: northern region, western region, and southern region, right? Now, among these varieties, as you know, the mango which are grown in northern India, North Indian variety, they are generally alternate bearer. And mostly the South Indian variety, they are generally regular bearer. Now the hybrid program was started in India during 1942. The objective of the mango breeding was that get a variety which is regular bearer, number 1. Number 2, attractive color, good size and shape, and have good taste. That's why that particular variety shows good demand in the market. That's why hybrid varieties development program was started. Hybrid variety. So, in case of northern Indian variety, the number 1 variety is Doseri, we have discussed last class. And in South Indian variety, the number 1 variety is Nilam. So initially, the cross between Doseri and Nilam. So, and to conduct this fruit breeding program, started in different fruit research stations, FRS, that is, different fruit research stations, which were established by ICR. These fruit research stations were established with an aim to develop good variety, develop new technology regarding management practices, suitable pesticides, suitable fungicides against pest and disease, and to find out the exact nutrient requirement of the plant. So, the hybrid varieties program was started, and first variety, that is known as Muhammad Bahar, that was developed Bombay Green. It is a northern Indian variety, cross with Kalapedi. And this was done in Fruit Research Station, Sabur, Bihar. At the same time, another breeding program was started where the name was Prabha Shankar. The parentage were same. Bombay cross with Talapedi. And that was in Futsal Station, Kodur in Andhra Pradesh. Kodur in Andhra Pradesh. Then different breeding program was started in different research center. Then another variety, that is Neludrine, Nilam cross with Himayuddin. Nilesan developed from Nilam cross with Bodesan. Nilgua, Nilam cross with Malgua. Nileswari, Nilam cross with Duseri. Next is Ratna, that is Nilam cross with Alfonso. Alfonso, it is a good variety, export variety from Maharashtra. Then Sindhu, that is developed. Ratna, because in case of Ratna, the required characteristics were not obtained. Hence, the Ratna was again crossed with Alfonso. And now this Sindhu variety, it is having a significant characteristics because this is the only mango variety which is seedless. Seedless means there is a presence of endocarp without any ovule, and that is hardly 6 gram weight only. That's why this is the seedless variety. Then another program was started in IRI, Pusa Institute, during 1972, and the varieties were released in 81, and that is Nilam cross with Doseri, developed in IARI, Pusa Institute, New Delhi. The variety is Mallika having a strong tendency towards regular bearing under North Indian conditions. Strong tendency towards regular bearing. And the fruits are medium sized to slightly bigger, but not so tasty. Hence, reciprocal cross was done and developed a good variety, Amrapali, that is Doseri cross with Nilam. In first variety, always you know in breeding program, the first variety is considered as a female. Nilam was female, Doseri was male. Then Dusari was female, Nilam is male. Now this is a very good variety. Have four important characters. Four important characters of Amrapali. Number one, highly regular variety. Highly regular variety. Every year there will be flowering and fruiting. Number two, distinctly dwarf variety. Distinctly dwarf, very short height plants. Distinctly dwarf. Number three, that precocity, early bearer. Precocity, the variety is precocity in nature, that is early bearer. That means if you go for grafting, then after third year, but second year, that means from third year onwards there is a flowering and fruiting. And you will find in arid conditions, 4 years old grafted Amrapali plant produces more than 50 fruits. And number four, highly nutritious. Highly nutritious in terms of highest vitamin A content. And this is the variety where the vitamin A content is 16,860 international units as beta carotenoids. Then another varieties developed by IIHR, Bangalore, Indian Institute of Horticultural Research. They have, because there was a problem, I have mentioned again Alfonso, exported variety, exported cultivar. But there is some problem, because we have discussed in the last class, Alfonso, small to medium sized fruits, attractive color, attractive yellow color. Pulp is golden orange color, golden orange yellow color. And the pulp is non-fibrous and having a very pleasant taste. And that raw taste of the raw pulp remains same in the processed products. When you prepare the squash jam jelly, after boiling the pulp, the original raw flavor of the pulp remain in the processed products. That's why it is accepted outside India. But there is a problem appears, that is spongy tissue problem. We will discuss in details. Spongy tissue problem means there is a fruit, this is a fruit, so you will find some of the portion of the pulps become hard, tasteless, colorless, and that portions remain green. That's why the amount of Alfonso export was reduced. That's why the IIHR, they have start the breeding program to develop the variety. Main object was to develop that main two objectives, that the food should be similar to Alfonso, and number two, free from spongy tissue problem. Then they have released Orca Punet, developed from Alfonso cross with Baganpoli. Then next is Orca Oruna, reciprocal cross. Baganpoli has female plant, cross with Alfonso as male plant. Then Orca on mall, that is Alfonso cross with Janardhan Prasad, another good variety from Western India. Now these Orca punnet, Orca aruna and Orca anmal, mostly the first two, punnet and aruna, they are completely free from spongy tissue problem. Flavor is similar to Alfonso, taste is similar, more or less similar, and nowadays these are also exported along with Alfonso, because in original Alfonso variety, management techniques are improved. That is also one of the another two factors, that harvesting at the late maturity stage. That may be one cause in Alfonso. Another cause, deficiency of calcium nutrient. So the mango growers, with the help of the scientists, improve the culture management practices to remove the Alfonso, spongy tissue problem from Alfonso variety. Now these are the good varieties. Now few varieties, they are also developed in some research stations, that is, and some varieties are still in the pipeline, not yet released. Manjira, that is Nilam cross with Romani. Then PKM no. 1, that is Swarnarekha, South Indian variety, cross with Nilam. Then Nilfonso, that was also earlier variety, Nilam cross with Alfonso. Then there is another variety, that is Niranjan. It is not a hybrid, it is a selection. Selection means when there are 1,000 plants of the same characteristics planted or sown, then you will find, due to some climatic conditions, not always, sometimes, 1 or 2 plants out of 1,000 plants may show different characters. And you will find that different characters are generally desired characters of the breeders. Earlier the plant was tall, bearing was late, production is not high, quality is not excellent. So in that case you will find plant becomes dwarf, spreading habit is less, bearing nature is more, early bearer, and the quality is good. Then immediately that plant is separated from the other plants. Then allowed to grow properly and ripen the fruits, then collect the fruits, extract the seeds, then sow. In case of pulse crops, vegetable crops, cereal crops, oilseed crops, these are seasonal, hardly for lasting, life cycle is 3 to 4 months. Then immediately collect the seeds and again sow, going for sowing. But it is a fruit crops, so you have to wait for minimum 5 years to produce fruits. Then you harvest the fruits, extract the seeds, sowed in the nursery. Plant will grow. Then again it will take 5, 6, 7 years to produce. And you have to compare the characteristics of the new plants with the original plants. If the similar characters, then again you have to grow for another several years, another 4, 5 years, to study whether the same characters are exist in the plants for future generation or not. If it is similar characteristics are found in future generations for 4, 5 years, means that plants produce the stable characters. Then that can be released. So immediately you cannot release it. Then you have to give all the description of the plant. Leaf characters, stem characters, flower characters, fruit characters, plant height, spreading, canopy area, all these things. depth of roots, spreading roots depth. Then you have to, with the photograph of the flowers, plants and fruits, seed size, seed shapes, with photograph of seeds, fruits, pulp, then send to the State Variety Release Committee. Then they will scrutinize. Then they will pass it. Then it will send to the Central Variety Release Committee. Then it is centrally released that, yes, this is the new variety released from such and such university or by some scientist. Now they have permit the particular organizations to name the variety. At the time it was in the code number. And the scientists or the organizations, then with discussion, they may release the variety with a name. So in that case, it is a selection. So in case of fruit crops, which are generally bear after 5, 6, 7 years of planting, it is very difficult. It takes minimum 15 years for develop a new variety.